All right, good evening, advanced human anatomy and physiology students. In section 9.3, there is quite a bit to do in this section. I will try and split it up, most likely into two separate sections for us for taking notes, because it is quite a bit. So basically what we are going to do is there are 5 objectives or learning outcomes for this section alone. The first one is to be able to describe the neural control of skeletal muscle contraction. Then you are to identify the major events of skeletal muscle fiber contraction. Next, list the energy sources for skeletal muscle fiber contraction. Describe oxygen debt. And lastly, describe how a muscle may become fatigued. So those are the 5 learning outcomes for section 9.3 alone. So what do we see here? Skeletal muscle contraction. Basically, contraction results from a movement within the myofibrils in which the actin and myosin filaments slide past one another, shortening the sarcomeres. So what do we have here? If you look here, this is a neuromuscular junction, abbreviated NMJ, neuromuscular junction. And basically what happens is you have the stimulation of a skeletal muscle cell. So at the neuromuscular junction, this is going to be the site where a motor nerve ending, or what we call a nerve fiber, is going to be in contact with a skeletal muscle fiber. So this is where the two meet, a nerve fiber and a skeletal muscle fiber. The area where they meet, which you can see here, this here would represent the nerve fiber, down here would be the skeletal muscle fiber. That area or space between the two is called the synapse or the synaptic cleft. So in order for a skeletal muscle to contract, its fibers must first be stimulated by that motor neuron. So the motor neuron is the nerve itself. You could see a figure of this in your textbook on page 298. It's figure 9.8. We also this neuromuscular junction is also known as a myo-neural junction. It is the site where an axon and the muscle fiber meet, and these are the individual parts. So the things that you're going to hear interacting here in this neuromuscular junction is you're going to hear about the motor neuron, the motor end plate, so that would be down in here. And you can see the figure here. The synaptic cleft is that space in between the two. You have the synaptic vesicles. Those synaptic vesicles are going to be the carriers of the neurotransmitter. And then you have the neurotransmitter themselves. So a motor neuron, as written there, it is defined as the nerve cell that stimulates the muscle cell. Neurotransmitters those are going to be the chemical substance that is released from the motor ending or the fiber. So at the end of the nerve, you're going to release these neurotransmitters, and that is going to cause a stimulation of the sarcolemma of the muscle fiber. So basically the plasma membrane of that muscle fiber is going to be stimulated, and the neurotransmitter there is acetylcholine, acetylcholine. Acetylcholine is the neurotransmitter, and it is also abbreviated capital A, capital C, lowercase h, ACH. The motor unit, one motor neuron and many skeletal muscle fibers makes up a motor unit. The motor end plate is the specific part of a skeletal muscle fiber's sarcolemma that is going to be directly beneath that neuromuscular junction. So you can see that right here, neuromuscular junction under high magnification. You can see the axon of the motor nerve, the motor end plate, the skeletal muscle fibers. Here you can see it again, a little bit higher contraction. So what is the stimulus for contraction itself? So ultimately, here you can see acetylcholine, the nerve impulse cause release of acetylcholine from synaptic vesicles. So the function of skeletal muscle is to move bones of the skeleton under voluntary control. Contraction of a skeletal muscle is a complex interaction of several cellular and chemical components. The final result from this is the movement whereby actin and myosin filaments are going to slide past one another. Accordingly, the muscle fiber is going to shorten and it pulls on its attachments. So the process begins, the process itself occurs when a motor impulse is initiated by the brain. It's going to travel down the spinal cord into a motor neuron, so that would be the nerve, which then branches into many motor nerve fiber endings. Each motor nerve fiber is going to extend into the motor plate end of the skeletal muscle fiber, forming a neuromuscular junction. When the motor impulse reaches the end of the motor nerve fiber, the membrane is going to be depolarized, and it's going to be depolarized from negative 70 millivolts to negative 55 millivolts. What happens? At this point, when you depolarize the membrane, you're going to open up gated ion channels, and calcium ions are going to rush in to the motor nerve fiber, and the neurotransmitter acetylcholine is going to be released into that neuromuscular junction. And that process that releases it is called exocytosis. So acetylcholine is going to diffuse across the neuromuscular junction, and it's going to stimulate and depolarize the motor end plate, or the sarcolemma, the nerve fiber cell membrane, of the skeletal muscle fiber from negative 100 millivolts to negative 70 millivolts. And what happens is the muscle impulse begins to travel over the surface of that skeletal muscle fiber and deep into that muscle fiber by means of the transverse tubules. So the muscle impulse eventually reaches the sarcoplasmic reticulum and those cisternae. Now we set up the excitation-contraction coupling mechanism. So here's where we're going to now see the interplay of the actin and myosin fibers. So excitation-contraction coupling, and then there's another form called the sliding filament model for muscle contraction. So in this form, the excitation-contraction coupling, the muscle impulse reaches the sarcoplasmic reticulum, and it's going to release those calcium ions into the sarcoplasm of the muscle fiber. Then, the calcium binds to troponin, moving tropomyosin and exposing the myosin binding sites on the actin filaments. Again, calcium binds to troponin, it's going to move tropomyosin and expose the myosin binding sites on the actin filaments. Now you could have linkages, or these cross bridges form between the actin and myosin. And when that happens, the muscle fibers shortens and contraction occurs. So it's what I just said there is kind of summarized on the side here, and you could see this in this diagram, which is also illustrated in your textbook. So that's the excitation-contraction coupling. So here you can see skeletal muscle high magnification. You can see the nuclei. You can see where those A bands are. You can see where the I bands are. sarcomeres. So remember that the A band is the dark area overlapping the thick and thin filaments. The I bands are those light, thin filaments. All right? Now, here is the sliding filament model of muscle contraction. And this is the more widely accepted, most popular theory concerning the contraction of muscles. The sliding filament model of muscle contraction, basically it was proposed by Hugh Huxley in 1954. It states that muscle contraction involves the sliding movement of thin filaments, which are actin, and the thick filaments, which are myosin. And you can see those illustrated here in the diagram, so the thick and the thin filaments. All right? And also take a look here. You have your Z line. You have your A band. You have the sarcomere itself. Okay? So now listen to what happens here. What's going to happen is sliding is going to continue until the overlapping between the thin and thick filaments is complete. So in a relaxed muscle cell, overlapping of thick and thin filaments is only slightly. That is, you get striations. However, this changes in muscles during contraction itself. So what then happens? The role of all this with muscle contraction. The distance between the Z line of the sarcomeres will decrease. The distance between the Z lines of a sarcomeres is going to decrease. The I bands, which are the light bands, shorten and disappear. The A bands move closer together, but do not diminish in length. All right. So the distance between the Z lines of the sarcomeres is going to decrease. Again, the I bands or the light bands shorten and disappear. And the A bands move close together but do not diminish in length. So relaxed, contracting, fully contracted. So what happened there? Basically, in a resting muscle cell, so we'll go back up here to the relaxed. In a resting muscle cell, that is, in the absence of calcium ions, tropomyosin blocks or inhibits the myosin binding sites on actin. Check this out on page 300, figure 9.9, illustration 1. And then when calcium ions are present, calcium binds to troponin, causing a conformational change. That's a change in form in the troponin complex, which allows for three things to happen. 1, tropomyosin to move. 2, it opens or exposes the myosin binding sites on actin. And 3, this results in the interaction between the active sites on actin and the heads, or the cross bridges of myosin. All right? And that you can see illustrated in figure 2. Now, the last thing is this cross-bridge cycling. So the cross-bridge cycling, what's going on in there? Figure 9.9 on page 300 illustrates this. When the calcium ions are present, the myosin binding sites on actin become exposed. That cross bridge attaches two things. ATP breaks down and is going to provide energy to cock the myosin head. Secondly, the cocked myosin head is going to attach to the exposed actin binding sites. So once tropomyosin moves, you expose those actin binding sites. ATP is going to cock the myosin head back, and it's going to allow that to attach itself to those binding sites. The cross bridge, which is the binding of the myosin head, springs from that cocked position and pulls on that actin filament. All right? Now the cross bridge breaks. ATP binds to the cross bridge but is not yet broken down. The myosin heads are released from actin, and as long as calcium ions and ATP are present, this stepwise walking continues until the muscle fiber has fully become contracted. So you can see the steps there outlined, that step process in the diagram. I think we'll stop there for today. That was quite a bit. Next we'll go on to relaxation energy sources and look at oxygen debt and oxygen supply. Thank you, everyone. Have a nice day.